Good afternoon, everyone. Thank you for being present here, and thank you to IEEE Epson for giving me this opportunity to present my work. I'll be talking about a sensitive and highly specific DNA-based impedimetric detection of Ralstonia Solanacearum in infected potato tubers without any PCR amplification. I just recently defended my PhD under the supervision of Professor Maryam, Shojaei, and Professor Rhea. Glad my professor is here with us for the talk today. I will quickly go to the objective of the work, a little bit on the literature, summary, experiments, and we'll discuss my results. So the objective is to develop a DNA-based biosensor to detect this bacteria called as Ralstonia Solanacearum using electrochemical impedance spectroscopy. And how can we apply this sensor, which is developed for real infected detection, and I use potato tubers as the disease sample for detecting this disease in potatoes. So what is causing plant diseases? A healthy plant can get infected via various ways. We have biotic as well as abiotic factors. What we were looking at is the biotic factors in which we have pathogens, which are disease-causing microbes, and they can be of various types like bacteria, fungi, nematodes. Ralstonia solanacearum is a gram-negative bacteria. It is soil-borne, naturally present, and is known to infect more than 200 different host plants, especially solanaceous crops. Why is it important to detect plant pathogens? Because crop pathogens can reduce the yield and the quality of the agricultural production, major occupation being in agriculture. It is very important if there is a technique by which we can diseases prior, so that we can have a lot of economical save and also save the crop for a better yield and production. Okay, and there are various traditional methods for detecting plant pathogen. The most gold standard techniques are microscopic visualization, polymerase chain reaction, and enzyme-linked immunosorbent assays. All of these techniques are laboratory techniques, require skilled labor, sophisticated instruments, and have certain limitations. To overcome the limitations of the traditional gold standard methods, there is a demand and a rise in biosensing platforms in the agricultural sector. So what is a biosensor? A biosensor has various elements. We look at the analyte, we look at the biorecognition element, and you can have various signal transducers. Here, the analyte, what we were interested in was the pathogen, Ralstonia solanacearum. Biorecognition element is a component which is very specific and can bind to the analyte. It can be of various types. You can have DNA, you can have an antibody, antigen, enzyme, substrate, lectins. What I worked on was a DNA-based sensor, and same way you can have various signal transducer principles. You can have optical, mass-based, magnetic, electrochemical, and the technique what we worked on was the electrochemical impedance spectroscopy. As I mentioned, there are various biorecognition elements. All of them have various advantages and disadvantages. We have DNA, lectins, antibody, nucleic acids, bacteriophages. Nucleic acids can be DNA and RNA. The advantage of using a DNA-based sensor is DNA is thermostable. It can withstand high temperature, and we can easily procure a DNA very specific to a particular target. And also we can bring about certain chemical modifications on DNA to enable a much sensitive and a faster detection. For example, a DNA molecule when we procure, we can also tag it with some fluorescent tags if you are looking for some fluorescent detection, or we can have even some thiol groups attached to it for better bonding with your electrode surface. I will be talking about electrochemical impedance spectroscopy as a signal transducer principle. What is EIS? The bulk and interfacial properties of the electrode system can be understood and quantitatively determined using electrochemical impedance spectroscopy. They can be of two types. You can have a ferritic as well as a non-ferritic approach. Ferritic reaction makes use of redox ions, and so that would need you to add certain additional reagents and not making it much simpler. Whereas a non-ferritic approach will not measure the kinetics of the reaction, but will be able to measure if there is any molecular layer organization or change in capacitance. And here you can have your buffers and simple solutions for which you can run the experiments without any redox ions or require any other additional reagent preparations. Ralstonia Solanacearum, as I also mentioned, that it's a gram-negative soil-borne bacteria. It infects various host plants. In fact, very important ones which we commonly consume, like potatoes, tomatoes, chilies, eggplants. This bacteria is present normally in the soil and does not require any additional conditions for grow. It can easily grow and proliferate. The major disease what this bacteria causes is known as bacterial wilting. That's the first sign and symptom which you'll see the drooping of the leaves down. That's called as wilting. How does it spread the infection is it colonizes to the roots and then it blocks the xylem, blocking the transport of water, and then it leads to eventually wilting of the leaves. For this work, we took collaboration with Indian Council of Agricultural Research, ICAR in Old Goa itself, with Dr. Ramesh, who is the principal scientist of the plant pathology department. The objective of this collaboration was before we actually do the sampling and studying of the DNA-based sensor, we need to first understand the microbiology of the bacteria. So I gained a lot of importance and I gained a lot of experience and a lot of insights on how this bacteria can cause disease, what are the conditions which we need to take into mind before we can actually work with developing a sensor for this kind of bacteria. I have a short clip, but if the time permits, I'll definitely go back and show it to you all. So this is the DNA probe. It's called as LPX-C4. It was a very specific probe and 30 nucleotides. It was thiolated, procured commercially. The reason for using a thiol functional group was we can enable efficient covalent bonding with the gold electrodes, which served as my platform for detection. Gold IDs, gold interdigitated electrodes, were the platform for developing this DNA-based sensor. These interdigitated electrodes were fabricated on a glass wafer micro-sized device by our project collaborators, FBK. So we have a project group in which we had FBK team from Italy, which were part of this team who worked on fabricating the devices. These devices were shipped to us, and then we had to work on how can we functionalize or how can we make it the best approach for bringing about functionalization and utilizing it for appropriate EIS measurements. The instrument used for the EIS measurements was a potential stat. We ran an impedance spectra from 0.1 to 1 megahertz at a very low amplitude of 5 millivolt because we do not want the voltage to disrupt the biomolecule organization. Initially, we made use of a simple random circuit just to understand what is going on the surface of the electrode, and later we also did a complex modeling to understand the geometry of IDE playing a role with the impedance spectrum. This is a general schematic of the work. Gold IDs which were given to us, we made sure that it is sufficiently cleansed and all the properties of the gold ID are matched for appropriate EIS measurement, which involved a number of measurements like fixing the volume, what is required, or how much is the averaging of the impedance data required for a good run, what is the storage condition of the functionalized ID, how much of the environment can affect, so how good an EIS can be run if it's done in an infrared case. So all of these parameters were first studied and fixed. Then the DNA probe, which I mentioned, LPX-C4, was procured. A method for functionalizing the gold ID with the DNA probe was finalized to ensure that it's successfully functionalized on ID. So this gave me the biofunctionalized ID. And then this functionalized ID was tested with N number of Ralstonia Solanacearum target, which served as a standard measurement, and then utilized this biofunctionalized ID also to detect infected samples, like I will be talking about potato samples here. Biofunctionalized ID was a two-step procedure. The IDs were thoroughly cleansed and functionalized with the LPX probe, and the unwanted sites were blocked with methylphenol. After functionalizing the ID, we had to make sure that the ID has been successfully functionalized with the probe. So a number of physical as well as electrochemical measurements were taken. Contact angle measurements, FTIR, indicating the peaks of DNA. These were some of the characteristic measurements which were done on the ID surface. EIS was also served as a platform to doing the electrochemical characterization. All of these measurements were performed in PPS because it was a non-ferritic approach. So if you take a bare ID and just drop the sample volume, what I used was 10 microliters. If you just drop 10 microliters and we run a PPS, that gives me my blank. Say the same ID after functionalizing, if I run with PBS, I should get a different spectrum. And that is what I observed, that the DNA, single-stranded DNA, will functionalize on the IDE, will block the surface for the ions to reach, and therefore you'll see a much increased impedance. And further blocking with MCH will also give a much higher impedance because MCH being having a very low dielectric. Then the functionalized ID was tested with the target strengths. Different concentrations were tested. So if a single stranded DNA probe gets a complementary target DNA, and if the DNA hybridization takes place, the conformation of DNA changes, and this conformation can be detected using EIS measurements. So a single stranded probe, once it gets its partner, it will form like rigid rods, which will again create space for the ions to come down. And that type of difference is what we could capture and measure. So more the DNA, target DNA present in a sample, more is the hybridization and more is the drop of impedance. Number of experiments were run to finalize the concentration. We could detect up to 0.1 nanogram per microliter concentration of target DNA. This was then modeled. I will quickly just go to my slides now. We also had to check with the mismatch studies. Yes, the DNA probe is binding to Ralstonia, but what if it also gives false positive results with other bacteria? So the sensor was tested with other commonly found soil microbes like Bacillus, Pseudomonas, Microbacter, and we saw that the developed sensor is very sensitive and specific to only Ralstonia. There were no changes in impedance with uncomplementary strands of DNA. Stability studies was another factor which was carried out. Cyclic voltammetry was carried out to make sure that all of this is happening in a non-redox fashion. There is no redox ion, so you will not see any redox peaks in your CV. Finally, we had to apply this sensor with infected potato tubers. Some 60 numbers of infected potato tubers from market, local regions of Goa itself were collected. They were sliced and they will look for certain browning around the vascular region. So if you see this browning, yes, it indicates there is some kind of infection, but is it Ralstonia or is it a secondary infection? We need to see. So from this bunch of suspects and this bunch of potatoes where there were no visual symptoms, but what if it has been contaminated with Ralstonia were tested. And to make sure that a sensor is not giving any false positive result, all the positive samples were also tested using the gold standard technique. That is microscopic visualization, the microscopic plating, PCR, agarose gel electrophoresis, all of this was done to confirm. And we were very glad to see that 3 positive samples out of 60 gave a impedance spectra indicating there is DNA hybridization. The 3 samples also gave positive for plating on very specific media, that is CPG plus CZC, what was used for Ralstonia. Yes, I'm done. Future work is we would require more rigorous field studies and also test other host plants. Look for getting into a prototype. Look for how we can take this technology to developing a sensor which we can detect for real field studies. And these are some of the references and work. Special thanks to all my supervisors, mentors, guidance I got throughout my PhD. And thank you.